Welcome back, everybody, for another deep dive. This time, we're taking a really in-depth look at Pseudomonas and Burkholderia.
Two biggies.
Definitely two biggies in the medical microbiology world. So if you're listening to this and you're in that field, well, you're in for a treat.
Absolutely.
So we're going to be working our way through the second edition of Principles and Practice of Clinical Bacteriology by Gillespie and Hockey. It's a really comprehensive resource. So much good stuff in this book.
It really is. It really is a really good book.
So let's get started, shall we?
Yeah, let's jump in.
I always found it fascinating how Pseudomonas was first discovered in blue pus from soldiers.
Oh, yeah, the blue pus. It's a pretty striking image, isn't it? And that blue pigment, pyocyanin, is a real giveaway even before you get to the biochemical tests.
So that's Pseudomonas aeruginosa.
The Pseudomonas aeruginosa.
The star of the show.
Yes.
So tell me about Pseudomonas aeruginosa. What are the key things?
Well, it's a gram-negative rod. It's got a single polar flagellum, so it's motile. And as you said, it's got that amazing pigment, pyocyanin, that blue one. And it's also got fluorescein, which is yellow.
Oh, I see. So it's a bit of a combination.
Yeah, it's a little bit of a combination. And that gives the cultures this really distinctive blue-green color.
Amazing. And this little bugger can metabolize a pretty wide range of organic compounds, right?
Absolutely. It can metabolize a whole range of different things. It uses an oxidative pathway for its glucose metabolism. Yeah, it's quite versatile.
So that's probably why it can live in so many different places.
Exactly.
And what about temperature? Does it like it warm or cold?
It likes it nice and warm. Around 35 degrees Celsius is its happy place.
Oh, okay. So it's kind of like a human.
Yeah, a little bit like a human, but there's one key difference. It can't grow at 4 degrees Celsius.
Oh.
Yeah, and that's how you can distinguish it from some of its close relatives like Pseudomonas putida and Pseudomonas fluorescens, which can tolerate the cold a little bit better.
Wow, that's a good tip for the lab.
It is a good tip for the lab.
Now let's move on to another tricky one. The Burkholderia cepacia complex. This one was always a real challenge to identify back in my studies.
It still is. Because it's not actually one bacteria, it's a whole complex of 9 genetically distinct but phenotypically very similar bacteria.
So they're like siblings.
A little bit like siblings, but they have different DNA, kind of like identical twins, but with different DNA. We call them genomovars.
Genomovars. That's a new word for me.
Well, a genomovar is basically a group of bacteria that share similar observable characteristics but have distinct genomic differences. And this makes identification really tricky because their biochemical reactions can vary quite a lot.
So how do we pin them down?
Well, they're slender rods and they're motile, and they prefer a temperature range of about 25 to 35 degrees Celsius. And their colonies, well, they come in a whole kaleidoscope of colors.
Oh, really?
Yeah, you can see grayish white, yellow, reddish purple, even a deep brown.
Sounds like a party in a petri dish.
It can be. But there are some biochemical tests that can help us. For example, they don't produce ammonia from arginine. They are positive for lysine and ornithine decarboxylase. And they have a slow oxidase reaction. And some of them even produce a melanin-like pigment when they're grown on tyrosine agar.
So we really need to be paying close attention to all of those little details.
You really do. It's detective work.
All right, let's move on to Burkholderia pseudomalae. This one I know is known for causing milioidosis, which can be a really serious infection.
Burkholderia pseudomalae is a fascinating one. It prefers a temperature of 37 degrees Celsius, and its colonies can be smooth or wrinkled. And the wrinkled form, well, it gets even more wrinkled when there's glycerol around.
Fascinating. And what about that smell? I've heard that it's quite distinctive.
Ah, yes, the infamous musty odor. It's a helpful clue, but I wouldn't recommend going out of your way to get a whiff.
Definitely not. And biochemically speaking?
Biochemically, it's positive for glucose oxidation, arginine deamination, and it can break down a variety of substances. It's quite versatile.
Sounds like it can really hold its own out in the world. Now, let's talk about a species that's not as common, but still important to know about Burkholderia malae.
Ah, Burkholderia malae. It's an interesting one, primarily an animal pathogen these days, mainly affecting horses. Thankfully, it's incredibly rare.
So it doesn't pose much of a threat to humans these days?
Not really, no.
But we should still know about it, right?
Absolutely. It's always good to be aware. It forms straight or slightly curved rods, and sometimes you can even see them in these arrangements that look like Chinese letters. And it's non-motile, unlike some of its relatives. And no pigment.
So it's not as flashy as Pseudomonas aeruginosa.
Not quite. Biochemically, it's catalase positive, but its oxidase reaction can be a bit variable. It reduces nitrate, but not nitrate, and it can be a bit unpredictable with gelatin hydrolysis, urease, and lecithinase tests.
So it likes to keep us guessing. Now, before we move on to the pathogenicity of these organisms, are there any other species that our listeners should be aware of?
The book also mentions a few other players like Burkholderia gladioli, Stenotrophomonas multifila, Ralstonia picaci, Bravundamonas species, Delphtia acidivorans, and Pandorea species.
Quite a mouthful.
It is quite a mouthful, isn't it? But they're all opportunistic pathogens. They don't usually cause problems in healthy people, but they can cause infections in people with compromised immune systems or underlying health conditions.
So it's like they're waiting to have the right opportunity to strike.
Exactly. And they've been popping up more and more in clinical specimens and hospital environments.
So it sounds like we need to be prepared for anything that might show up in the lab.
Absolutely. We never know what we're going to find.
Right. All right, now let's dive into what makes these organisms such formidable foes, their pathogenicity. Let's start with Pseudomonas aeruginosa, a true master of virulence.
Oh, yes. Pseudomonas aeruginosa is a real pro at causing trouble. It's got a whole arsenal of virulence factors: exotoxin A, phospholipase C, proteases, pyocyanin, which we talked about earlier, but it's also a toxin as well as a pigment. It's got pulley, flagella, and lipopolysaccharide.
Wow, it's like a one-bacterium army.
Pretty much. But interestingly, even with all of that firepower, it's not considered to have high intrinsic virulence. It's often described as an opportunistic pathogen.
So it needs a little help to really cause trouble.
Yeah, exactly. It needs to exploit a weakness in the host, maybe a weakened immune system or a large number of bacteria entering the body or a break in the body's defenses like a wound or a medical device. But when it does get that opportunity, watch out.
It's a reminder that context is everything when it comes to infection.
Absolutely. Now, one of the coolest things about Pseudomonas aeruginosa's pathogenicity is its type 3 secretion system.
Oh, yes, I remember learning about that. It's like a bacterial syringe, right?
You got it. It's a molecular syringe that allows Pseudomonas aeruginosa to inject toxins directly into host cells.
Like a microscopic hit squad.
Exactly. These toxins interfere with all sorts of cellular processes, leading to cell death and tissue damage. And on top of that, don't forget about biofilms.
Yes, biofilms. Those bacterial fortresses. They always cause so much trouble in the clinic, especially with chronic infections.
They're a nightmare. Biofilms are basically communities of bacteria that are encased in a slimy matrix. And for Pseudomonas aeruginosa, a key component of that matrix is alginate, which is a mucoid exopolysaccharide that gives the biofilm its slimy texture.
So they build their own protective shield.
Exactly. And it helps them stick together and to surfaces, which makes it really hard for antibiotics and the immune system to reach them.
So it's like they're hiding out in their own little fortress, safe from attack.
Pretty much.
That's fascinating and scary.
It is.
Now let's talk about Burkholderia cepacia complex. What makes it so pathogenic?
Well, one of the big things about the Burkholderia cepacia complex is that it can survive inside host cells.
Inside the cell, so not just hanging out on the surface.
Yeah, they're not just surface dwellers. They can actually invade and live inside cells. And this intracellular survival is a key to their success. Well, it allows them to evade the host's immune system and some antibiotics.
It's like they're playing hide and seek with their defenses.
Exactly. They're very good at hiding. And the virulence of Burkholderia cepacia complex, it can vary a lot depending on the genomovar. Some genomovars are a lot more pathogenic than others, especially for those cystic fibrosis patients we talked about.
Right.
For example, there's one called Burkholderia sinocepacia, and it can be really aggressive. It can cause a rapid decline in lung function in CF patients.
So it's not just about identifying the species, it's about knowing the specific genomovar.
Absolutely. It's really important to get that level of detail. Oh, and there's another interesting thing about Burkholderia cepacia complex that's still being studied. They can invade and survive inside amoebae.
Amoebae, like those little single-celled organisms that live in water and soil.
That's right.
Wow, so they can even hide out in amoebae.
Yep, and it makes you wonder if those amoebae could act as a reservoir for Burkholderia cepacia complex in the environment.
That's amazing. It's like these bacteria are playing a whole other game that we're just beginning to understand.
It really is. It's quite incredible.
Okay, last but not least, let's talk about Burkholderia pseudomolae. What do we know about its pathogenicity?
Well, Burkholderia pseudomolae can produce a whole bunch of toxins and enzymes, but we're still figuring out exactly what role they play in causing disease. And like Burkholderia cepacia complex, It can survive inside macrophages.
Macrophages. Those are immune cells, right?
Right. They're the big eater cells of the immune system. They're supposed to engulf and destroy bacteria, but Burkholderia pseudomalae can actually live inside them.
That's sneaky.
It is. And we think that this intracellular survival is a big part of why melioidosis can be so chronic and why it tends to relapse. The bacteria can hide out inside these cells and then reemerge when the conditions are right.
So it's not a quick fight, it's a long game.
It's definitely a long game.
Wow. And there's a link to diabetes, right?
Yeah, that's right. People with diabetes are more likely to get melioidosis, and they tend to have more severe cases. And we think it might be because Burkholderia pseudomalae has a specific binding site for human insulin.
Wow, that's fascinating. So it's not just about the bacteria, it's about the host's individual characteristics too.
Exactly. And that's really important to keep in mind when we're trying to understand infectious diseases.
That's a good reminder for all of us. Okay, I think that's a good place to wrap up part 1 of our deep dive into Pseudomonas and Burkholderia.
Yeah, I think so too.
We've covered a lot of ground already. We've talked about the characteristics of these organisms, their pathogenicity, and how they interact with their hosts.
It's been a great discussion so far.
Yeah,
I
yeah.
can't wait to dive into the clinical features, diagnosis, and treatment of these infections in the next parts.
Me too. Stay tuned, everybody. We'll be back soon.
Yeah, we'll be back soon. Welcome back, everyone, to our deep dive into Pseudomonas and Burkholderia.
So last time we left off talking about how sneaky those organisms are.
Yeah, real masters of hide and seek.
They really are. So now I'm curious, what does our book say about preventing and controlling these infections, especially in a healthcare setting? Because that seems like where they really thrive.
It really does. And, you know, it's funny, the book really emphasizes those basic infection control practices that we always talk about, you know, hand hygiene and sterilizing equipment.
Yeah.
But it's so important, especially with Pseudomonas aeruginosa.
It's easy to forget about the basics when there's so much exciting new stuff happening in microbiology.
I know, but honestly, those simple things, they can make such a difference. Making sure everything's properly disinfected, keeping a close eye on things like water systems for contamination.
That's good for Pseudomonas aeruginosa. What about Burkholderia cepacia complex? What does it say about that?
Well, for Burkholderia cepacia complex, the book stresses that it's really important to separate infected patients from those who aren't, especially in cystic fibrosis centers.
Oh, right, because those patients are already so vulnerable.
Exactly. And you also got to be really careful with medical devices and solutions, making sure everything's sterile and handled correctly.
So no cutting corner.
No cutting corners.
Okay.
Because some strains of Burkholderia cepacia complex, especially that Burkholderia sanocepacia we talked about, it can spread so easily between CF patients.
So you got to be extra careful.
Extra careful. Now, for Burkholderia pseudomalae, it's a little different, right? Because it's more of an environmental threat.
Right, so how do you prevent that?
Well, the book says it all comes down to education and awareness, making sure people who live in or travel to those endemic areas know about melioidosis and how to protect themselves.
So like wearing protective clothing and being careful around soil and water.
Exactly, especially during the rainy season when there's more Burkholderia pseudomalae around. So it's all about giving people the information they need to stay safe.
Right, right. So those are the preventative measures. What about treatment? What are some of the big challenges there? I mean, we've already talked a bit about antibiotic resistance, so I imagine that's a big one.
It is a big one. And, you know, the book doesn't sugarcoat it. It's pretty upfront about the challenges, especially with Pseudomonas aeruginosa, because it's resistant to so many antibiotics.
So what do you do?
A lot of times you have to use combination therapy, you know, hit it with multiple drugs at once, like beta-lactams, aminoglycosides, fluoroquinolones, even polymyxins sometimes.
So it's a bit of a balancing act, trying to kill the bacteria without causing too much collateral damage.
It is. And we always have to be mindful of the risk of driving further resistance. We don't want to make the problem worse.
Right, right. And what about Burkholderia cepacia complex? Is it just as tricky?
Oh, it can be even worse. The book says that treating Burkholderia cepacia complex infections is really complex because they're resistant to so many antibiotics. So you really got to tailor the treatment to each patient. You know, look at their individual situation, the specific strain they have, and what it's susceptible to.
So no easy answers there.
No easy answers. And sometimes even the susceptibility testing can be misleading. You think you found the right antibiotic, but it doesn't work in the patient, so it's a real challenge.
So it really is a case-by-case basis. What about Burkholderia pseudomalae? What does the book say about treating melioidosis?
For severe melioidosis, the book recommends intravenous ceftazidime, often with cotrimoxazole.
Ceftazidime. Okay. And why that one?
Well, it's a beta-lactam antibiotic, and it's got good activity against Burkholderia pseudomallei. And the cotrimoxazole, that helps broaden the coverage just in case there's some resistance lurking. But, of course, you always want to base your treatment decisions on the specific patient and what their susceptibility testing shows.
Right, right. Always personalize it. And we can't forget about the risk of relapse with melioidosis. How do you deal with that?
The book suggests prolonged oral eradication therapy. Basically, you give them a combination of antibiotics for an extended period to try and wipe out any lingering bacteria and prevent them from coming back.
So it's a long-term commitment.
It is. It's not a quick fix. But it's often the best way to prevent those relapses.
Right. So it sounds like with all of these infections, antibiotic stewardship is really important.
It's absolutely critical. The book talks a lot about using antibiotics wisely, only when they're really needed. Choosing the right antibiotic based on susceptibility testing, making sure patients finish their entire course of treatment.
All the things we preach day in and day out.
Exactly, because we got to keep those antibiotics working. We can't afford to lose them to resistance.
Right, right. So is there anything else that's important for managing these infections besides just the antibiotics?
Yeah, the book emphasizes that it's not just about the drugs. It's about supportive care, too. Making sure the patient is comfortable, well-hydrated, getting good nutrition, managing any complications that might come up, like sepsis or respiratory failure.
So it's a holistic approach.
Exactly. And for patients with chronic infections, like those CF patients with Pseudomonas aeruginosa, they might need long-term management strategies, like airway clearance techniques to help them get rid of mucus and bacteria from their lungs, and inhaled antibiotics to deliver the medication right where it's needed.
So it's a long-term battle. Now, before we jump into those exam questions, you mentioned some other Pseudomonas species earlier. Are there any specific treatment challenges with those less common species that we should be aware of?
That's a great question. Yeah, they might not be as common, but those other species can still cause a lot of trouble. And often it's because they're resistant to multiple antibiotics.
Can you give me an example?
Sure. Stenotrophomonas multifida is a good one. It's naturally resistant to a lot of the antibiotics that we commonly use, including carbapenems.
Carbapenems, those are like our big guns.
I know, but Stenotrophomonas multifida has this enzyme, L1 metallobeta-lactamase.
A metallobeta-lactamase, that's a tough one.
It is. It breaks down carbapenems, so they're basically useless, so it makes treating these infections really tricky. Sometimes you got to use less common antibiotics, and those might not be as effective, or they might have more side effects.
So it's a real challenge.
It is, so it's really important to stay up to date on those resistance patterns.
Right. Always be learning. Well, I think I'm ready for those exam questions now.
All right, let's test your knowledge. All right, question number 1. Which pigment is unique to Pseudomonas aeruginosa and gives it that characteristic blue-green color?
Oh, I know this one. It's pyocyanin, that beautiful blue pigment that always makes Pseudomonas stand out on a plate.
You got it. And remember, pyocyanin isn't just a pigment, it's also a toxin. It actually contributes to the bacteria's ability to damage host tissues. So it's playing a double role there.
Sneaky little bugger. All right, hit me with question number 2.
Okay. What is the most common mode of transmission for Burkholderia cepacia complex among cystic fibrosis patients?
This is a clinically important one. I think it's person-to-person transmission, because those patients are often in close contact, and Burkholderia cepacia complex can spread through respiratory droplets, contact, or even contaminated surfaces.
You are absolutely correct. Person-to-person transmission is a major concern in CF centers, which is why those infection control measures we talked about earlier are so important. Separating infected patients, cleaning equipment carefully, good hand hygiene, all of that.
It all adds up. Okay, I'm ready for question number 3.
Burkholderia pseudomallei causes which disease known for its varied presentation and potential for relapse?
Oh, that's got to be melioidosis. That was a real chameleon. It can look like so many different things. And that relapse, that's what makes it so tough to treat. It can hide out in the body and come back later.
You're exactly right. Melioidosis is a tricky one, both to diagnose and to treat. All right, 2 more to go.
Bring it on.
Question number 4. Which antibiotic is considered the treatment of choice for severe melioidosis?
Okay, let me think. For severe cases, I believe it's intravenous ceftazidime, usually given with cotrimoxazole as well. Is that right?
You nailed it. Ceftazidime is a good choice for Burkholderia pseudomallei, and the cotrimoxazole helps to broaden the coverage. But as always, you want to tailor the treatment to the individual patient and what their susceptibility testing shows.
Always personalize it. Okay, last question.
All right. What is the primary mechanism by which Stenotrophomonas multifida exhibits resistance to imipenem?
Stenotrophomonas multifida, that's a tough one.
Yeah.
But I think I remember. It produces a metallobeta-lactamase, which can break down carbapenems like imipenem.
That's exactly right. That metallobeta-lactamase is what makes Stenotrophomonas multifida so resistant to so many of our antibiotics. It's a real challenge to treat.
It's a good reminder that antibiotic resistance is a constant battle.
It really is. Well, congratulations, you aced all the questions.
Thanks. I have to say I learned a lot from our deep dive today and from this fantastic book.
Me too.
And to our listeners, we hope you learned something new as well. We hope this deep dive gave you a fresh perspective on Pseudomonas and Burkholderia and that you'll be better equipped to deal with them in the lab and in the clinic.
And remember those basic infection control practices. Hand hygiene, sterilizing equipment, all of that. They might seem simple, but they're really important.
Absolutely. So until next time, stay curious, stay vigilant, and keep those lab coats clean.